Enzymes in recombinant DNA technology. Enzymes are molecular tools that perform precise functions in manipulating DNA and RNA. These are essential for cutting, joining, modifying, copying, and amplifying genetic material. 1. Restriction enzymes, restriction endonucleases, function. They cut DNA at specific recognition sequences, usually palindromic, producing blunt or sticky ends. Types. Type 1. Cleave DNA randomly away from the recognition site. Type 2. Cut DNA within or close to the recognition site, most commonly used in rDNA technology. Type 3. Cut at a short distance from the recognition site. Examples: enzyme source organism recognition site type of cut E. coli GAATTC sticky ends hind III Haemophilus influenzae AAGCTT sticky ends Bomi Bacillus amyloliquefaciens GGATCC sticky ends Alui Arthrobacter luteus AGCT blunt end SMAI Serratia marcescens CCCGGG blunt ends. Small diamond 2. DNA ligase. Function. It joins DNA fragments by catalyzing the formation of phosphodiester bonds between the 3' O and 5' phosphate ends of DNA strands. Examples: T4 DNA ligase, from T4 bacteriophage, most commonly used ligase. E. coli DNA ligase. Works well with NAD instead of ATP. Small diamond 3. DNA polymerases. Function. They synthesize new DNA strands from a DNA template. Types and uses. Enzyme function and application. TOC polymerase heat-stable enzyme used in PCR, from Thermus aquaticus. DNA polymerase I fills gaps after RNA primer removal in DNA synthesis. Clenau fragment a portion of DNA pol I used in blunt-end cloning. PFU polymerase high-fidelity polymerase used in high-accuracy PCR. Small diamond 4. Reverse transcriptase. Function. Converts mRNA into complementary DNA, cDNA. Source. Retroviruses, e.g., Maloney-Murine leukemia virus MMLV, used in RT-PCR, cDNA library construction. Small diamond 5. Alkaline phosphatase. Function. Removes phosphate groups from the 5' ends of DNA to prevent self-ligation of vectors. Sources: calf intestinal alkaline phosphatase, CIAP, shrimp alkaline phosphatase, SAP. Small diamond 6. Polynucleotide kinase, PNIC. Function. Adds phosphate groups to the 5' ends of DNA or RNA, allowing them to be ligated. Source: T4 polynucleotide kinase, from T4 phage. 7. RNase and DNase. RNase. Degrades RNA, used during DNA isolation. DNase. Degrades DNA, used during RNA purification. 8. Terminal transferase function. Adds nucleotides randomly at the 3' end of DNA strands. Use. Tail addition for ligation, creating homopolymer tails for cloning. Summary table. Enzyme type function. Key examples: restriction enzymes cut DNA at specific sequences. EcoRI. Hind III DNA ligase join DNA fragments. T4 DNA ligase. DNA polymerases synthesize DNA strands. TOC. PFU. Clenau reverse transcriptase converts RNA to cDNA. MMLVRT alkaline phosphatase prevents vector self-ligation. CIAP. SAP polynucleotide kinase adds phosphate to DNA, RNA. T4 PNIC RNase. DNase degrades RNA or DNA RNase A. DNase I terminal transferase adds nucleotides to 3' DNA ends. TDT.